The broadcast is now starting. All attendees are in listen-only mode.
Hi, welcome to this iPathwayGuide overview webinar. I'm Cordelia Zeraldo, the product manager at Advaita Bioinformatics, and I'll be hosting today. Before we get started, I just have a few quick orders of business. The first one is an audio and visual check. If you can see and hear this presentation, could you please just give us a quick chat in either the question window or the chat window of your GoToWebinar control panel. Looks like we've got confirmation, so we'll keep going. Thanks, everyone. The next order of business is to say that this webinar is being recorded, and we will send a link out to everyone who registered after we have completed. So if for some reason you get interrupted, you can come back later and pick up right where you left off. Please use the questions tab in the control panel to ask any questions you have throughout the presentation, and we'll save time at the end for a discussion. And lastly, there will be some announcements about upcoming events, so stay tuned to the end for that. All right, introducing today's speaker. Today's speaker is Mr. Andrew Olson, the Vice President of Business Development here at Advaita. Mr. Olson holds an MBA from the University of Michigan, Ross School of Business, and is a serial entrepreneur with a business development background in a number of industry settings. As a former university tech transfer principal, Mr. Olson has helped commercialize a number of technologies. He has extensive experience in managing technology licenses, joint ventures, mergers, acquisitions, and divestitures, and has served as a mentor to many entrepreneurs. At Advaita, he has been intimately involved with many aspects of the commercialization and development of iPathwayGuide. Through this, he has become an expert in all aspects of iPathwayGuide, from uploading and analyzing data to understanding the algorithms powering the results. Okay, let's begin.
Okay, thank you, Cordelia. Sorry about the visual difficulties there for the beginning there, but looks like we got that all straightened out now. So again, my name is Andrew Olson, so I'll be walking you through the overview here for iPathwayGuide. And if I can just get this to work, there we go. So today's agenda, we're going to just this is just a high-level overview of what iPathwayGuide can do in terms of looking at your gene or protein expression data. We'll compare the standard methods for analyzing that kind of data and contrast that with how iPathwayGuide handles the data. And then we'll give a live demonstration of iPathwayGuide, which should give you kind of a sense of how to navigate and get started with it. And then at the end, like Cordelia said, we'll have some Q&A and then some announcements as well. So let's just dive into that. All right, so Advaita, we're a fairly small team, but and we've been around for about 4 years, but in that time, we've amassed over 9,000 registered users to the iPathwayGuide and our iVariant Guide application. We have a number of customers from a variety of institutions, from academia and pharma and various research foundations. And it's my pleasure to announce that we recently signed on the National Cancer Institute. So I know that we have a few members from NCI that are on here and other members from NIH that are on here. And there's an announcement about some future activities with that towards the end. So the core functions of iPathwayGuide are to identify your significant DE genes. We predict the activity of microRNAs. That's based on a gene expression signature or protein expression signature. Then we provide gene ontology analysis, and we have some advanced algorithms that help you identify the specific terms that are most interesting based on your data. And then, of course, pathway analysis, where we also give you the ability to model drugs, microRNAs, and variants directly on the pathways. And then we also do enriched diseases, and then we have a meta-analysis function. And I'll talk about each one of these in greater detail during the live demonstration. So there's a lot of features and benefits that you can get from iPathwayGuide. I'm not going to go through every single one of these things, but I think some of the key things that I hear from our customers are that our advanced methods really help them because they don't have to slog through a bunch of false positive results, and I'll show you how that works in just a second. Our meta-analysis, which allows you to quickly compare and contrast different analyses and identify either common or unique traits. We have an open sharing model so that if you have an experiment that you want to share with a collaborator or a colleague, you can openly share that. We don't require that person to have a license. They only need to have a free account to our application. And then the user interface is also, I think, one of the things that people talk about a lot is it's easy and intuitive to use. Okay, so when we talk about the bioinformatics pipeline here, we go from instrument and raw reads all the way through to pathway analysis. I just want to make sure that everybody understands that iPathwayGuide is really focused on this tertiary analysis portion, the pathway analysis. The only exception to that is that we do accept for certain platforms in the Affymetrix line raw cell files, which equate to a sample each. But otherwise, for your RNA-seq experiments, there are generally defined pipelines that either your informatics core or another service that you may be using will get you to differential expression. And iPathwayGuide picks up from that point on, just so you understand where we play in that entire ecosystem. So let's talk about some of the classical analysis approaches to pathway analysis and how to interpret this differential expression data. So the most common one that everybody is probably familiar with is what's called overrepresentation analysis, or oftentimes it's referred to as enrichment analysis. You hear about a pathway is enriched. And what they mean is when they look at that data, they just look at the number of differentially expressed genes on that pathway, and they assess whether that's more or less than what they would have expected across that entire data set, and they score it with a p-value. The benefit of this approach is that it's really simple. All you need is a list of genes. But the cons of that, though, are that it assumes that the genes are independent, and we know precisely that they are not independent. I mean, that's exactly what the pathway is there to describe, is how gene A influences gene B, and gene B in turn influences gene C. It's a nice, quick and dirty type of analysis, but it is prone to providing a lot of false positives. So an improvement on that is generally what's called functional class scoring. If you've heard of gene set enrichment analysis, or GSEA, that's a type of functional class scoring. And it is more accurate than your standard enrichment approach, but it still presumes that all the genes are independent. And so, again, you are left with a number of false positives. So another approach, though, is to look at the pathway topology, or how those genes are connected, and assess how perturbed a particular pathway is based on its topology. So in that approach, you consider the role and the position of each gene, and you can kind of fill in gaps in between your measurements because the pathway is there to describe the relationships between all of the genes that are on that pathway. The downside on this approach, though, is that it's computationally intensive and it requires a little bit more data. So rather than just a list of genes, you also need associated fold changes and p-values. So what we do, though, is we take the best of both worlds and we combine them in a method called impact analysis, where we take overrepresentation analysis, and then we also take the topology-based analysis, and combining those two, we get two forms of evidence that allow us to assess the significance of each pathway. So there's a reference here at the bottom here. The method that we are using here comes from Soren Dragishi's lab from Wayne State University, and this was published in Genome Research in 2007. But essentially what we do is we look at all of the relationships between every single gene on every single pathway, and whether that's a one-to-one relationship as described by the purple dot and the blue dot, or if it's a one-to-many relationship, or possibly even a many-to-one. The point here is that we look at all of the types of interactions, the number of them, the direction, and we assess that at every single point between every single gene on every single pathway. And that's really different from every other commercial application that's available out there. So when we combine these two different types of approaches, this enrichment approach and the perturbation approach, we end up with this two-way evidence plot. So on the horizontal axis, you can see we use the minus log of the p-value that comes from the overrepresentation analysis. That means that the more significant pathways are going to be on the right end of the scale. Similarly, on the vertical axis, we use the minus log of the p-value that comes from our perturbation analysis, and therefore the more significant ones are at the top of this axis. Ideally, when you combine those, you would want to find those pathways that have two forms of evidence are in this kind of top right quadrant. And that is your kind of your signal for, hey, this is something that you should really be looking at. So let me give you just a very quick and brief example of how enrichment analysis differs from impact analysis. This is a real world type of example. So here we have the insulin signaling pathway. I know it's a little bit difficult to see in your screens, but I've highlighted here a single gene. This is the insulin signaling receptor. And if we just pretend, for example, that this is our one significant gene on this pathway, enrichment analysis is going to look at this and say, well, I have one significant gene out of a possible, let's call it 50 other genes that exist on this pathway. It doesn't care whether this gene is an integral part of this pathway or not. It simply looks at the ratio of significant genes versus non-significant genes. And so in this case, the enrichment analysis is going to say, well, okay, might be reasonably important or maybe not. Our impact analysis, though, is going to look at the specific gene, and it's going to see that, hey, this is the main entry point to this particular system. And you can see it has many, many connections, and so if this gene, for example, were to be knocked out, then you would be able to see that the rest of the pathway would also be knocked out. And so from that, it's able to assess how significant this particular pathway is. Let's take a look at another pathway. In this case, this is the adherence junction pathway. And in this case, we have that same insulin signaling receptor. And in this case, again, we have, let's say, one DE gene out of maybe 50 genes that are on this pathway. And so enrichment is going to score this pathway very similarly to the previous pathway. But our impact analysis method is going to look at this, and it's going to see that in this particular pathway, this receptor plays a much less significant role, doesn't influence the downstream genes nearly as much as in the other pathway. And so it would score this pathway less significant than the previous one. So I hope that kind of gives you an idea of how this method is able to tease out which pathways are more significant and more relevant to your data set than enrichment analysis alone. Okay, so let's just talk about briefly, and I'll demonstrate this in the demo here in just a second, but for us, data, our input data is the entire list of genes, not just the top 100 or 200 genes that you might think are significant. We want to see the entire list of genes, and that's so we can also assess the background of this list, and that's for statistical purposes. Along with that list of genes, though, we also want to see the associated fold changes between the two conditions that you're looking at and their corresponding p-values. And we recommend that you use corrected p-values. And we support currently human, mouse, and rat. We've been getting a lot of requests for other model organisms, and currently we aren't supporting those other organisms, but we do have a number of customers that are doing that type of analysis using orthologs, so that might be an opportunity for you as well. We support most of the major file formats that are out there. As I mentioned, we do AFI-cell files for certain Affymetrix platforms. CuffDiff, DE-seq, EdgeR are kind of the three common workflows that you get from RNA-seq. If you happen to be a Jump Genomics customer, we support their platform. If you have a bioinformatician that is familiar with some of the packages in R, we support the Lima package. We also support the Geo to R platform that you can sometimes process DE genes in the gene expression omnibus. We support NSolver for nanostring, SciEx for protein for proteomics, and then of course we have this custom tab delimited file. So basically, as long as you have those three things, the gene symbol, fold change, and p-value, if you put that in a text file, which Excel can output for you, you're up and running. And we'll have protein ID. We'll have a protein ID workflow coming out very soon. Okay, I'm going to demonstrate this in a second, so we'll just skip over that. Just in case we do run out of time, I did want to mention the meta-analysis. The meta-analysis is a premium feature, and it allows you to build up or compare up to 5 data sets, and you can even combine mRNA data with proteomics data, and it allows you to quickly identify those unique or common traits, and even see if there's a change in expression. So it also works really well for if you're doing a drug study for different therapeutics, or even a dose study or a time course study, and stuff like that. So it's very useful. And we do have some webinars from last year that hit on some of those things, and I'll show you how you can get to those later. Oh yeah, the printable summary is another feature. I'll show that to you as well. But each data set that is purchased comes with a printable summary. It's about an 18 to 20 page report that's automatically generated for each data set. That is a nice way to kind of put some language around the analysis, and you can attach it as additional information in, say, a manuscript or your grant submission. So it's a great feature. We find a lot of life scientists really enjoy that because it helps them to understand and convey what the analysis identified. Okay, let's dive into the actual application demonstration. So if you haven't, from our website, advaitabio.com, you can register for a free account. Just go to the top right corner, hit register. And then once you've activated your account, you'll get an email. Please make sure you click on the activation link in that email, and then you should have full access here to go into the application. So here we're going to be showing the iPathwayGuide account. So we hit login there. It's going to ask us for our email and our password. And when you're presented with your account, you should have these exact same demo data sets in your account when you come in. So if you want to later on, you can follow along. As Cordelia mentioned, we'll be sending out a recording of this, so you don't have to necessarily do it right now, but you can kind of follow along. So this is our dashboard. This is the list of the available analyses that we can look at. Let me start off by showing how you would get your data into the application. It's really simple. You just hit Analyze New Experiment in the top right corner. Choose your organism. So we have human, mouse, and rat here. And then the file format. We default to CUFDIF, but you can choose from any one of the other ones that I mentioned previously. And I'll actually show a CUFDIF file because that seems to be one of the more common types that get submitted to iPathwayGuide. And then you simply choose your file. In this case, here's my demo data set. Oh, we actually have demo data sets available on our website. Just go to the FAQ and you can download sample data if you want to try doing this yourself. Then we click Upload. And it just takes a couple seconds for this to go up into the cloud and process. It might be slightly delayed here because we're also broadcasting on this webinar. Here we go. So you just give your analysis a title and then a brief description. And then you have, in this case, this CUFDIF file actually has 3 available contrasts that we can look at. And we could actually analyze all 3 of them if we wanted to. And when we do that, it'll actually construct a meta-analysis by default on these 3. And you'll see how that works a little bit later. From the CUFDIF file, if our application can parse out the contrast labels, it will automatically do that, but you're welcome to edit those contrast labels here. So maybe you just want to call this control instead of the GMCSF. And then here we see the number of genes that were identified in that analysis, and we have our default threshold. So we use 0.6 for the fold change, and then the adjusted p-value is 0.05. These are fully adjustable, so if you would rather use a 1.5 fold change, you can do that. Just make sure you hit view and apply to apply that threshold. And you'll get actually a preview of the volcano plot for that gene expression. You can even see some of the genes that are going to be included in that analysis. I'll just go back to our 0.6 for our fold change, and we find that that seems to work best, but it really depends on your particular experiment. That's why we give you the ability to customize it. So now that I've done that, it's updated, and we can see that we have 503 DE genes that are going to be analyzed in the context of these almost 11,000 genes in the data set. When you're satisfied, just hit Analyze Data, and it'll zoom off, and in usually about 15 to 20 minutes, you'll get an automated email saying that your analysis is done. So I'm going to spare us that wait, and so we'll dive into one of these demo data sets that we have here. All of these come from public data sets. You can just punch in the GSE number in your Google or search engine, and you'll get the link to the paper and the data set that these were based off of. So I'm going to show you this top one here. This is a classic treated versus non-treated type of data set. So in this case, the researchers were looking to identify what the impact a particular miRNA would have on gene expression in a cell line. Before I forget, when you go into the analysis, right next to the data set or to the title of the data set, you'll see this little plus sign at the top. If you click that, you can get more information. You'll see actually the title that you gave it and the report description. You can edit those. You can also edit the contrast labels if by chance maybe you chose the wrong contrast label. But more importantly, I think, is the list of data sources that went into the analysis. And you can see the dates. This is actually off of an older version of the data set, but or of our database. We update our database about every 6 months. We'll be coming out with an update to it probably sometime just after the first of the year. And if an updated database is available, you can actually, the button will appear here and tell you that, hey, you can update your data set if you want. So there's some version control there for your data, and we'll preserve the old results. So you can always update it to the latest and greatest, but we'll catalog each of the previous results for you. Okay. So the summary page that you're presented with is quite that. So on the left, we have the input. So this is our volcano plot. We see what the organism was. We have about 1,200 significant differentially expressed genes out of almost 21,000 total genes. You can see what the thresholds were used to construct the volcano plot and the DE genes. And then on the right side is our output. So we have top enriched pathways, top pathways that are overrepresented and also perturbed. And then we have our predicted microRNA analysis. We have our gene ontology analysis for the 3 domains: biological processes, molecular functions, and cellular components. And then we have top enriched diseases. If you remember, I also mentioned about this printable summary. It comes up very quickly. You just click on that printable summary, and you'll get a full list of kind of a background of what went into the analysis. You have the full report of what the methods were that were used to construct the analysis, and then the results with raw p-values as well as FDR and Bonferroni corrected p-values, along with the top 5 results from each category with captions and figures and so on and so forth. So this goes on for quite a bit. You're welcome to explore these on your own, and I highly encourage you to give that a shot. Okay, let's go into the Genes tab. So we'll work across from left to right along the top here. So the Genes tab is really the central point of the analysis that you'll be looking at. Here you can, if you have a particular gene that you're interested in, most researchers have a favorite gene or something that they're studying, you can search for that, pull it up, see where it is in your volcano plot, find out more information about it. also see which different biological processes or pathways, drugs, etc., are annotated to that particular gene. This is all very interactive. If you wanted to download the graph, you can save that as a PNG or an SVG file. You can also save the table that's down below it as a comma-separated file. So, of course, this is disabled for trial reports, but any purchase report has full download capabilities. Another really cool feature is this little camera icon, and you'll see them kind of sprinkled throughout the application. If you click on any one of those camera icons, you'll be presented, whoops, sorry, I meant to just pause that. You'll be presented with a short 1 to 2 minute video that gives you just a little bit of additional information about that particular part of the application. So you don't have to sit through another 1 hour long webinar trying to find out what that one part of that application is about. So I encourage you to use the in-app videos to learn a little bit more, and they're usually at the very top of each section within the application. So just a little tip there for you. Okay, let's move on to the microRNA analysis. This is where the real analysis begins. People sometimes get confused. They think that we're actually analyzing microRNA expression in this analysis, but it's actually the opposite. We're looking at mRNA, and from the mRNA expression signature, or actually it could also be protein expression signature, we are predicting the presence or the activity of a particular microRNA. So in this case here, we have the graph in the top left corner with the ranked results, and then we have the associated table below it. By default, we do not apply a correction factor, but they're easily applied. We don't hide them. We have Bonferroni, which is the more stringent of the two, or we have FDR. So I'll use Bonferroni in this case. And I don't know if you saw it, but the order doesn't change, but the p-values change. So in this case, now we are left with 4 significant results here. But this first one here is much more significant, 10 to the minus 4. And in this analysis, we've identified that miR542-3P is the most significant or probably active microRNA in this particular sample. And the way that we do that is by looking at the number of downregulated DE genes that are targeted by this particular microRNA, and then we compare versus all of the DE targets, and we compare that to all of the targets, not just the DE. So in this case, we had 45 out of 47 DE targets that are downregulated out of 187 versus 279 of all the known targets of this particular microRNA. And over here on the right, we have a little bit more details about this particular microRNA. We have link outs to more information about it. We have the stem loop diagram, but then we have the gene expression bar plot. And here we see there are a number of downregulated genes, and we have 2 that are upregulated. So all of these genes are significant in our data set and are targeted by this particular microRNA. The cool thing about this gene expression bar plot is that each one of these bars is a link to the Genes tab that gives you more information about that one. So I just clicked on ILK, and so it gives me more information about it, what it's kind of a summary of its function, some literature references, and other annotations for that particular gene. What's really cool about this microRNA analysis is that, if you remember this particular data set, the researchers were really looking to identify how a particular microRNA would impact gene expression. And so we've kind of gone the other way. Based on that gene expression, we identified that this miR542-3P, which is the one that these researchers used, is the most likely active microRNA in this particular sample. So our application, without knowing that ahead of time, was able to accurately choose and identify the microRNA that may be active in this. Okay, so let's go on to gene ontology analysis. So in gene ontology analysis, there are 3 domains: biological processes, molecular functions, and cellular components. They all function very similarly, but obviously the terms, the gene ontology term, describes a different type of function in this case. So gene ontology analysis, if you're not familiar with it, is a structured vocabulary that identifies and has genes annotated with each of the different processes, functions, or cellular components. And it is a hierarchical structure. I'm sorry, let me scroll down just a little bit further. And the root node will, in this case, I chose biological processes. All of the terms in this domain will end or terminate with biological process. So, and when I demonstrate this, it'll become a little bit more clear for you. But one of the inherent problems with gene ontology analysis is, and most researchers that I talk about this type of analysis with, they tend to roll their eyes because it's really not very specific, doesn't seem to lend them a lot of insight. Well, we've solved that problem here. And the way that we do that is, here we have our plot of the significant results. And the way that we read this is we start at the 12 o'clock position, we work counterclockwise, and as we get into the magenta and kind of purple areas, we get more and more significant. Now there's maybe 500 significant terms here, and that's really hard for any one person to slog through. So we have Bonferroni and FDR as correction factors, but those really are not appropriate in this type of analysis because these are not true independent measurements here. So we have elim and weight pruning, and this takes into account the hierarchical structure of the gene ontology and balances the specificity with the specific term and relevance to your data. So in this case, I'm going to choose elim pruning, which is the more stringent of the two. And you can quickly see that we've eliminated a lot of the magenta terms, and we're left with maybe only a couple here. We can go a step further and require that we have at least, let's call it 24 DE genes for each term for it to be included in our list. So instead of having several hundred, we've got probably, I don't know, I'm getting a guess, probably about 65 terms here. But the most significant one is much more significant than the other terms. So this is a great way to dive into certain things that may be very relevant for your particular data set. In this case, we've identified the negative regulation of cell proliferation as the most significant gene ontology term in this particular data set. And we have the relative count, so we have 79 DE genes out of 588 that are annotated to this particular process, and we have the corresponding p-value. On the right, we have a link out to the specific term and a little description about it, and that gene expression bar plot that we showed in the microRNA analysis as well. Down below, though, we have the ancestor chart. So as I mentioned before, they're all going to end in this particular domain with biological process. And then down below, from that, you can see down below how we get to this negative regulation of cell proliferation. There's a legend on the right here. And basically by highlighting each one of these terms, you can see how it highlights the different paths all the way up to biological processes. If there's one particular term that you're interested in, you can just click on the title or the Go number on it, and it'll collapse that tree to that particular term. I encourage you to use the in-app video if you want to learn a little bit more about it. Just click up here in the top right corner, and you'll get that video that explains a little bit more about how that gene ontology analysis works and how you can leverage that for your particular data. All right, let's move on to pathways. So if you remember from the slides, we use those two forms of evidence to assess each pathway. So on the horizontal axis, we have the p-value that comes from the enrichment analysis. And then on the vertical, we do our perturbation analysis, basically where we take the topology into account. And when we combine those two p-values, we get a single result, or I should say we get a single score for each pathway. In this case, we've identified the pathogenic E. coli infection pathway as the most significant pathway. Just to contrast this a little bit from enrichment analysis alone, if we were only using enrichment, so we would just be looking at it from the horizontal axis, this p53 signaling pathway would be our most significant, followed by biosynthesis of amino acids, followed by gap junction, and then finally we'd get to the E. coli infection pathway. So you would have had to go through 3 false positives before you would have gotten the most significant pathway in this case. We offer correction values. I suggest at least using an FDR at a minimum, and you can see how the scale adjusts here on the axis. And we still have our E. coli infection as our most significant pathway. On the right here is the specifics about this particular pathway. So in this case here, we have the E. coli pathway described here, and we see that we have a scale for our fold change that came from our experiment. So by default, that's what we're highlighting here on the pathway. So we've got a number of genes that are downregulated, and they're colored in blue, and we have one that is upregulated here. So these pathways come from KEGG, and many of these nodes on the pathway can be families of genes or complexes. And if you wanted to understand which genes were measured, you just simply hover over each one of the nodes, and you'll see which genes came from your experiment and what their relative expression was. We highlight the most significant in terms of fold change, absolute fold change, as the default color here. When we assess these pathways, remember, we also take the topology into account. So in this case, I'm just going to kind of focus in here on this section right here. So the pathway describes a relationship from this tubulin complex or to this tubulin family to these other downstream genes. So if we accept the pathway as a knowledge base of these relationships, we can take what was measured here and propagate it downstream. And that's what we do when we hit this accumulation button. So this is the downstream accumulated measurement or predicted fold change, if you will, of the downstream genes. Then we can look at the total perturbation. So this combines what was measured plus the downstream component, and it shows us what is predicted to be impacted in this particular pathway. By showing coherent cascades, what we look for is coherence between the data that was identified in our experiment and then also the relationships that exist in the pathway. When those two things are in agreement together, we highlight those with red arrows. And these serve as putative mechanisms in the pathway. And essentially, these are the areas that you should be looking at as a researcher. And these areas explain all of the measured changes in this particular system. And I'll show you a little bit more about this in just a second. But I wanted to make sure that I also showed you down below that we also have the gene expression bar plot that was measured from our experiment. We also have a box and whiskers plot. And many people are always are asking us, well, can you tell me if this pathway is activated or inhibited? This is kind of the key for that, but you have to take that into context as to how the pathway is described and what it is describing. But generally speaking, if you see that this box and whisker plot is on the negative side, that would indicate that that particular pathway is being inhibited. All right, now let's look at some details. So I'm going to click this plus button in the top right corner, and it's going to open up a panel here on the right that allows us to go into much greater depth. Again, there's a little video icon here for you to go through and understand a little bit more about some of these different things. Particularly, I think the part about the bootstrap diagram is very interesting, but I'm not, in the interest of time, I'm not going to go into that here, but please explore that on your own. If you remember from our microRNA analysis, we had identified this miR542 as being the most likely active microRNA. We can model that directly on the pathway and identify the target for that particular microRNA, or possibly targets. They may have more than one. In this case, this particular microRNA happens to target this tubulin B2B, which is identified right here in the table, and happens to be the source of this particular coherent cascade or this putative mechanism in this pathway. So if I were a researcher, I would be very interested in this particular section of the pathway, and I may have already identified a plausible hypothesis for why this particular section of this particular pathway happens to be impacted. So now I could set up a functional study to see if indeed this particular microRNA is affecting that, or maybe a drug or some other type of stimulant in this particular cell line that would allow me to validate or go a little bit further in my research. So let's go on to diseases. And quite clearly, this is not a disease data set, so take these results with a bit of a grain of salt. But the way that we look at these diseases is well, first of all, we use the International Classification of Diseases of 2010 to classify the various diseases. And the rings of this particular graph go from the most general to the most specific, from inside to outside. So here we have congenital malformations. Here we have neoplasms. We have endocrine disorders, diseases of the nervous system. And maybe this would give you an idea of something that you might want to investigate further, or give you some ideas about possible therapies or other things. So clicking on any one of those rings helps you focus your results or your interest in that area. And then as you go out further, it continues to get more and more specific. In this case here, this collagen myopathy is the disease that's been identified. We see the significant genes that are driving that particular disease as being significant, and you have more information about that here, and we have literature references, etc. If you want to zoom back out, you just click on the middle, and that takes you out to the starting point. Again, we've got these little camera icons. Please use those. Before I forget, because I usually forget, if you wanted to share your results, just click share in the top right corner. And in here, you can put in anyone that has an email. If they don't have an account, they'll be prompted to create an account. But you can control whether they have the ability to reshare. So if maybe you have a grad student or something like that, and you just want them to look at it, but maybe not do any further sharing, you can restrict that person's ability to reshare just by unchecking that. Once they show up here, you just click share and they'll get an automated email. All right, cool. So that's kind of the application looking at a single contrast. And I encourage you to kind of write your questions down if you have any questions about that. But while you're doing that, I'm also going to dive into a meta-analysis. And this is also available as a demo in your account when you register. So in this case, we're looking at, this again comes from a public data set in which the researchers wanted to understand some breast cancer subtypes. So they have triple negative versus healthy. We have HER2 versus healthy, luminal A, luminal B. And then at the bottom here, we have this meta-analysis. So clicking that meta-analysis brings up this ability to identify regions that may be of interest to you based on your criteria. So we have all of those contrasts here on the left, and they're all color-coded, so you can see that this kind of pinkish area is the triple negative versus healthy. And if you were just interested in those 706 DE genes that are potentially driving that phenotype, you would click on that region, and then the table down below is updated automatically with just those 706 genes. We can also build up custom tables by clicking these individual regions, or if we wanted to, we could just actually even click the entire bar here on the left. You can also clear the regions up there. If I wanted to look at the 521 genes that are driving or that are in common across all of these phenotypes, clicking that 521 builds that table very quickly for us. You can then further investigate any one of the genes that happen to be in that table just by simply clicking on that gene, and then we get more additional information about that particular gene. We have a little description, but then how the expression changed from one phenotype to the other, along with the specific values in the table next to it. And we also have some literature references below that. Where I think this particular meta-analysis becomes very, very powerful is when you're looking at, say, gene ontology analysis. So let me pull that up quickly here. And then remember, we always like to apply a correction factor. In this case, we're going to use the elim pruning. And if I wanted to identify any particular gene ontology analysis. I can take it from the table, I can click on it, I can rank it by p-value. So here we have the G1S transition term that is most significant in this triple negative contrast. But if I wanted to, I can also look at the rank. And this is where it becomes very powerful. So I clicked on it, there was a button here, it switches from rank to table view. And I can look at which things are significant. And here I'm looking at triple negative versus healthy. And so we have these in ranked order as from most significant to least significant. And I can quickly go through and I see I've got some DNA strand elongation, I've got DNA replication. So I've got some DNA type of processes that seem to be affected or enriched in triple negative and not so in the other contrasts. If I want to shift my focus, I just simply click on the header, and now I see that extracellular matrix or ECM is very significant in the other cancers, but less so in triple negative. If I wanted to, again, I think you get the idea, but you can click through and find anything that is of interest to you and the associated gene expression for those genes that are annotated to that particular term. It's also really powerful in the pathways analysis too. It functions very, very similarly. We'll apply a FDR correction factor, and I simply look at the rank diagram. And again, here we have DNA replication as being most significant in the triple negative versus healthy. And if I want to find out more information about those genes, I can start diving into it a little bit further. And when we do that, we start seeing here, in this case, we have MCM2, MCM3, MCM5, MCM6. We have a lot of these MCM genes that are significant and upregulated in the triple negative, but not significant in the other breast cancer subtypes. So these could potentially serve as plausible biomarkers for triple negative versus healthy, and clicking on any one of those genes gives me more information. So in this case, the one that I chose here kind of at random was most significant here in the triple negative versus healthy. It met our threshold criteria for full change in p-value, but when we look at the others, I probably should have prefaced this, we used a 2.0 threshold for the fold change in this particular experiment because I believe that's what the researchers did in their paper. But we see that the HER2, luminal A, and luminal B did not meet our threshold criteria. And you know it when it meets your criteria because it's in bold, and in this case, these are not bolded. So that's the meta-analysis, and that works across all the different analyses that we have here. If you have any questions or issues, just use this comments feature here in the top right corner. Cordelia and I generally get these things, and we're very responsive. If you ever have a question, just shoot us a question. We're happy to answer them. If we can't, we'll find the answer for you. And so I think with that kind of concludes the overall analysis of the application and how it can work. I'd like to now open it up for questions if you have any. Please use the dialog box in the control panel, and we'll get through as many as we can. If by chance we run out of time, we've only got about 10 minutes left in this presentation. I promise that either I or Cordelia will follow up with you after the webinar and hopefully address your questions directly. So while you're formulating those, I just wanted to let you know this is for our NIH and specifically NCI, CCR attendees here. We will be in Bethesda on December 19th for the Bioinformatics Training Education Program Workshop. That's a hands-on training, so if you want, you can bring your data, and we'll also have some sample data, but we'll be doing a hands-on all-day event of how to use iPathwayGuide and get the max out of that. And then because of the holidays, I don't think we're scheduling any webinars for December, but we are working on putting together a joint event with our friends at NanoString. We'll be doing a joint webinar sometime in January. We'll be making an announcement as soon as we have that date solidified. But we'll show you how you can use NSolver and how you can leverage that and get a meta-analysis out of that, and very similar to what we did today. But we'll be going into specifically NSolver data and the workflows, how to get that expression out of Ensolver. So with that, let's take some questions, Cordelia.
Okay, great. We have quite a few questions, so I'll just go for some of the more common ones. The first one is, can I use images from iPathwayGuide in a publication or even text? And if so, how should I cite iPathwayGuide?
Yes, you can. In fact, we highly encourage it. Just please check the end user license agreement. If you are using a trial account, you are not allowed to use that for publication purposes, but we do have pricing that really fits any person's budget. But actually, let me go from our website. You go to the support panel in the top right corner, if it works. I may have lost my web connection. Here we go, sorry. And from there, we have a number of FAQs, and I think right down here, question number 13, how should I cite iPathwayGuide? There's a little guide there that tells you how you should do that. And while we're here, I just wanted to point out we have a number of other resources here. We have some tutorials, we have some pre-recorded webinars here where we look at time course expression, comparing protein and mRNA expression, breast cancer subtypes, et cetera. So there's a number of use case scenarios that have been identified here in our support page for iPathwayGuide that you might be able to leverage.
Okay, great. The next set of questions was about the types of data you can analyze in iPathwayGuide. So if you could just talk about sort of the range of data types that you can input, and then specifically, can you use iPathwayGuide with microRNA expression data? And what about qPCR that has a small number of genes, like less than 500 genes in a data set?
Yeah, those are all good questions. So let me just go back into the application here. So from the Analyze New Experiment, you really get a sense of what we accept. So human, mouse, and rat for organisms. And then the file formats. So AFI cell files, again, look at our FAQ for the specific platforms that are supported. We don't support every single platform, but like the human HU133 2.0, I can't remember exactly the specific one, but that's one of the most common ones that it's out there. We support that, and some similarly for mouse and rat. CuffDiff, DE-seq, EdgeR, those are your typical RNA-seq workflows. But really, here I'll even show with a custom tab delimited file, if we choose that a text file that happens to have that, here it is. All you need to do is identify which column contains your gene symbol, which column contains your fold change, and which column contains your p-value, and you should be off and running. Now, the question about let's talk about microRNAs. No, we do not take microRNA data. Currently, this needs to be a gene symbol, not a microRNA symbol. And but as I mentioned in the slides, we will have was actually supposed to be released last week, but in the next week or two, we should have protein ID for your symbol, so you don't have to convert your protein IDs to gene symbols. So I'm sorry we don't support the microRNAs yet. We've had a lot of requests though of late, so you never know, it might be coming out soon. The other question, qPCR, there are some things that are unique for smaller data sets. In fact, we'll be covering that in our webinar with the NanoString people because their platform also is generally restricted to, I think, 770 genes. So we'll be identifying that, but you have to adjust some of the statistics accordingly, and or apply or provide some additional background genes for it. So we'll address that in that January webinar.
Okay. This person would like to get an idea of the proteins that are modified downstream of genes, and they would like to know if we can use meta-analysis to combine proteomic and transcriptomic data sets in order to do that.
Yeah, totally. That's an awesome question. Whoops, not in the slides. Over here. So again, I think it's right here. So we even have a pre-recorded webinar. It's on our support page, comparison of protein and mRNA expression profiles. Have a look at that. It should give you a good sense of how you can combine those. We've worked with some people from UCSF and SciEx as our proteomics partner, and we've done a number of posters and other studies looking at that specific question. So it's really quite fascinating. There's a lot of research that still needs to be done on some of that stuff, but it's super cool.
Great. So this question is about correction factors and what is the significance level that's used to determine significance after applying correction factors.
Oh.
And how are the correction factors applied? So how can we find information about that?
That's another good question. So let's look at going back into this particular analysis. So we use the traditional 0.05 as the significance threshold as for what is significant and what is not significant. So I'll pull up Pathways here. I think that will show it fairly well. So I'm going to just scroll down and focus on the table here. So by default, we have the raw p-values, and these are ranked in order from most significant to least significant. Again, the no correction factor. But let's apply a Bonferroni and see what happens. So here we can see the rank order generally does not change when you're applying these things. It actually shouldn't change. And so as we go down this particular list, I'm going to increase the number here. We can see that they're in green and bold up to 0.049, or actually it's 0.050. And then anything that's greater than that is not bolded. And so these would not be considered significant. So that's the significance part of it. How they are calculated and how they are applied, that's you can find out more information on that. I think we have something in our FAQ, but another great resource is this printable summary that's available in any one of the demo data sets, or if you happen to purchase a particular report or get a subscription, this would be automatically generated. We have the methods here, but then calculation for how we do the correction factors is contained in one of these figures. I'm sorry, I don't remember exactly, but I know it's in here. And so that's another great resource for you to look into.
Okay, follow up to that is if I have significant pathways before correction factors and no significant pathways after, what does that mean?
Wow, well, it can mean a couple things. If I'm not going to go into great detail, but you have to kind of look at your input data to assess if the input data itself was significant. And I can do an offline thing with you on that if you have a specific question on that. But generally what it means is that nothing was significant. And an answer of, hey, nothing has significantly changed is just as valuable as saying, hey, this particular pathway is highly impacted. So, and that's where our perturbation analysis really takes the cake on being able to assess that. Because now you have two forms of evidence that assess those pathways and not just an enrichment score. Enrichment, let's face it, it's a bag of genes. It doesn't consider if gene A is related to gene B. But, if gene A is your one significant gene and it happens to be, highly expressed or inhibited, then it's going to have a greater impact on that entire pathway. And enrichment just can't assess that. And so with our method, if there's nothing significant after applying a correction factor, I can reasonably say that there's nothing significant based on that data.
Okay, thanks. I think we have time for one last question. It's 2:59. And that is, first, is iPathwayGuide free? Or what are the licensing options for both academic or nonprofit and for-profit users?
That's a great question. In fact, this is going to be changing come January 1. So I highly encourage you, if you're interested, to contact us about some pricing and or different subscription options. But basically what we have, you can purchase an individual report. Usually most academics that are not interested in that. They don't want to be held back by the number of reports they want to analyze. So we have a 30-day subscription that's available for academics only. That currently is priced at $599. I believe that price is going to be going up in January. I don't know exactly how much. And then for annual subscriptions for academics, it starts at about $3,500, but it's super reasonable if you have a few users. It only goes up, I think, by a few $100s for each additional user on that, so it can be very, very economical. We even have some site licensing options that are available until the end of the year for academics. That basically a site license is about the price of one seat to Ingenuity's IPA. For commercial, contact us. Those prices are actually changing the most come January 1. Unfortunately, that's dictated by some of the databases that we use and the licenses that they provide to us. So I wish I had more control over it, but unfortunately I don't. But if you're interested, give us a call. We're happy to set up or create a custom quote for you and help you assess, get the right subscription for you.
Okay, I think that wraps it up.
Cool. All right, so this will be recorded. We'll post out a link to this in the next 24 hours or so. We'll even probably post it up on YouTube. So if you have, if you want to look at it there, we'll make sure you get a link to it. And if you have any questions, please let us know. Actually, I'm sorry, I forgot to put this up there. But you can go to our website, advaitabio.com, or hit us at info@advaitabio.com. Both Cordelia and I will get those emails, and we'll make sure to respond to you within a business day. So thank you very much, everyone. Have a great rest of November and December. And if we don't talk to you, we'll see you in the new year.